It gives me great pleasure today to welcome our speaker, Becky Pfedehirt. Did I say that right?
That is. I always told people that you pronounce my last name, the P is silent, like in Pfizer. And for most of my life, people would look at me like, what are you talking about? And now over the last couple of years, people are like, oh yeah, Pfizer. We all know Pfizer. So yeah.
The pandemic
helped me.
Yeah. And she's a partner with the A16Z bio investing team. And she focuses on early stage companies building tech platforms for therapeutic discovery and development. And so we'll be talking about this pretty soon. She has a very storied career from, let's see, before Andreessen Horowitz, you were at Amgen, where you led business development for therapeutic platform partnerships. Before that, you were a research scientist in Amgen R&D on cell and gene therapy innovation. She was also a postdoc in Genentech, and she holds a PhD in Molecular and Cellular Biology from UC Berkeley, and a bachelor's in biology from MIT. A lot there. We are thrilled to have you, Becky.
Thank you. I'm really thrilled to be here. This is super special, so thank you.
Please give a round of applause to our speaker. So the way I'm envisioning we do this is, we'll have about 40 minutes of questions that I've gotten from the community, and then about 20 minutes for the audience Q&A. So I think the best way to start off, or the best way to introduce someone, is actually to ask about their own experiences. And as I just read, you have a very interesting career starting from biology at MIT, as a scientist, as a postdoc, and then BD, and now at A16Z. So can you walk us through that journey? Because I feel like you might have met a lot of interesting people and experiences along the way.
Yeah, absolutely. So I was recently asked to give a talk at my alma mater at Berkeley about my career, and they were like, would you do this, and can you give us a title for the talk? And I had 30 seconds to move on to my next thing in the day, so I was like, sure. The joy of the unknown. And I say that because I truly think that one of the best parts, in my opinion, about a career in general, and I think this probably jives with what all of you seem to be interested in terms of wandering into new spaces and learning new things, is how joyful the unknown can be in life. And that might be the unknown of your career path, or that might be the unknown of just learning new things along the way and exploring in new spaces. So when I started out at Berkeley, I thought I wanted to be an academic faculty, as many people who start their PhD do. I quickly realized that wasn't what I wanted to do, and that was, I'd say, because of two things. I learned that I was someone who loved diversity in topics, probably like many of you, again, and successful academics oftentimes need to go really deep into one topic to be very successful there and publish well and become an expert. And I love to dabble and learn new things, and I've always felt like I had scientific ADD. As soon as I was working on one thing, I was like, oh, new shiny object, other new shiny object, So that was one thing. And then the other thing was I realized, I want to do important work that means something and that will contribute to the world. And there was a bunch of different ways that could actually end up looking at the end of the day. And I absolutely believe that academic work is very important, and most of the companies that we invest in and build are oftentimes based on academic work, but I wanted to be closer to that translation to something that actually reaches people. And so as I was trying to figure out what to do next, there were a variety of different options I could go into, but I realized that I still also wanted to be able to explore in science and to not take some R&D job where I was going to be told to develop some assay and follow some really boring project that had been laid out ahead of me. I still wanted the freedom to explore areas that I was interested in. And so exploring different options, I discovered that there was this amazing thing called the Genentech postdoc. And I still to this day tell many people who I talk to who are exploring, what do I want to do next? I want to further my scientific career, but I also want to learn what biopharma is like, that many companies in the biotech sector have postdoc programs, and there are these amazing opportunities to do both of what I said, to have freedom to explore and choose the directions you want to go in and play a bit in science, but also to have mentorship from folks who are deeply steeped in biotech, in advancing actual programs, and who are going to be supportive of your interest and career in that space. So I got to work at Genentech in some really interesting programs, mostly focused on trying to understand how post-translational modifications, so additions that are made to proteins, how that affects their role in disease biology and how that ultimately could be targeted as a therapeutic program. As I wrapped up my postdoc at Genentech, again, was kind of trying to figure out what to do next. I actually interviewed for a job at Invitae. I spread the bucket wide because I was like, one of the ways to learn what the options are just to like apply to a lot of jobs, have a lot of conversations with folks, figure out maybe this isn't something I want, maybe it is. Job postings sometimes are not at all what the actual job is. So I think not applying for something because the job posting sounds boring is probably not the right way to do it. And in fact, the job that I ended up taking was like the most boring job description I think I applied for. It was a job at Amgen in this technology development group. And I think the description said something like molecular biologist needed. And I was like, this is probably going to be really boring, but who knows? We'll see. In the interview I had with that hiring manager, I was like, what are you looking for? Because this is a very vague description. And he said, I'm looking for 3 things. I want to hire someone who's smarter than I am. I want to hire someone who gets along with my team. And I want to hire someone who's not afraid to try things and fail. And I was like, this is the job I want. Because it tells me that he's looking to surround himself with people who know things that he doesn't. He's not afraid of having team members under him who are actually, I don't know about if I was smarter than him, but who are interested in different areas. He builds a team who cares. He cares about culture. He cares about people working together as a team. And this is going to be a role where you have freedom to try things and fail. And like, what else could we want in life than the freedom to get paid to try things, and failure is okay as long as you're trying something hard. So I joined this team, got to work on some really cool projects, mostly related to trying to come up with new ways to be able to regulate cell and gene therapies, be able to engineer cells to do novel functions, be able to deliver therapies to specific cell types. But as part of this kind of skunkworks team, we were always looking at external innovation because we were this small team who was tasked at like envisioning the future and coming up with new technologies that could enable Amgen's drug development programs. And oftentimes the things we were working on, there were entire biotech companies with 50 people and $150 million in funding who were doing the same thing that like one of us was trying to do. Plus, because of my scientific ADD, I was always like, if I could work on many projects, that would be optimal. And the way I can work on many projects is by setting up partnerships with multiple different companies and then being the manager of all of that. And so I was always coming to our business development team saying, hey, have you heard of this cool company? Have you heard of this amazing academic work? Let's set up a partnership here. Let's set up a partnership there. I don't think before that I actually even knew what business development was. I thought it was like sales and marketing. And in some places it is sales and marketing. But interestingly, in biotech, business development is actually like scientific matchmaking. And it requires someone who understands the science at a very deep level, who can see scientific innovation A and scientific innovation B and be like, oh my God, together 1 + 1 = 10, and they actually solve problems that each other have. And then also someone who can communicate that both at a scientific level as well as at a very high executive level to folks who aren't scientists. And then also someone who can think creatively and say, okay, but how does this make sense business-wise? Why is it that these two companies, other than the scientific combination, why is it that this makes sense financially? How are both companies going to benefit at the end of the day? And so I ended up moving into a business development role at Amgen and did that for about 4 years and was really focused on looking for early stage innovation. So oftentimes companies that were at maybe like the seed, Series A, Series B stage, and trying to set up those collaborations between Amgen's R&D and these startup companies. But the reality is, at most biopharma companies, it is easier to in-license a $3 billion phase 3 clinical asset than it is to do a small early stage R&D collaboration. And there's many different reasons why that is we can go into. But I spent a lot of time being frustrated by the cool science I was seeing and the difficulty in actually doing something about it. And I was lucky enough, so to your point around kind of people you meet and relationships that happen along the way, I was lucky enough to have my office next to this amazing woman, Janice Nave, who led Amgen Ventures at the time. And Janice and I, spent a lot of time around, like, this was pre-COVID, so around the, coffee machines and coolers and whatever. And Janice was like, hey, these cool new things that you're discovering, what if we invest in them instead? And maybe over time we can develop a relationship and turn it into a business development deal. And so I started bringing more and more things to her. And I realized venture had never been a long-term career goal for me. But I realized venture is this incredible way to get to identify really promising early stage science and actually be able to advance it to a place where either the company can pursue that product themselves or that can be advanced via partnerships with biopharma. And so via a series of different introductions and conversations I had with folks, I ended up getting introduced to the team at Andreessen Horowitz. And all of you probably know this, but I get asked all the time, like, what is the difference between Andreessen Horowitz and A16Z? 16 letters between the A in Andreessen and the Z in Horowitz. Because imagine if if my email address was Rebecca Pferdehirt at AndreessenHorowitz.com, like, I would never get an email, which maybe would be a good thing. But anyways, as an aside, I met the folks at A16Z and I immediately was like, these are my people, this is my place, this is where I'm going to get to run and really do the things that I want to do with an incredible team of people who are very like-minded. So I've been there for about 2 years, and it's been a ton of fun.
Very interesting that, so was getting to a venture firm always in the plan when you started, or it just organically happened that way?
Yeah, so getting to a venture firm was never in the plan. That was, I still, when people ask me like what I do, my gut reaction is to say I'm a scientist, yes, because like fundamentally that's how I view myself. And I just think that this is an amazing way, back to like the original statement about having scientific ADD and liking to work in a lot of different places and getting to learn and meet amazing people. This is the way that I can do a lot of science with a lot of different people and impact many different areas rather than diving deep into one. And maybe someday I'll find one thing that I want to dive deep into and I'll start my own company. I don't know, but like for now, I love this. It's really fun.
Yeah. Thank you. Thank you for sharing that journey. So, joy of the unknown. I think that jives well with the South Park Commons community here. I like your term scientific ADD. I haven't heard that one before.
I coined it.
Yeah. Okay. Is it patented? Can I use it? Because
no, you can't. I'm sorry.
Okay.
Yeah.
I tried.
Okay. You would have a license though.
Yes.
Yeah.
Yes. I also love the fact that you've aligned your training as a scientist with your personality of scientific ADD in this case, but also like talking to people and betting on these early initiatives. One of the things that while we are going through this curriculum at TechBio that keeps coming up is biology is hard, biology is slow, science is slow, there's too much regulation. And I think it's far from true, not right now, because there are so many different things that are changing that biology maybe 20 years ago was slower, but it's scalable now. And the example I give is, you can read, write, and understand biology at scale now. Mm-hmm. So with all all these new cool technologies coming in, and where you sit at A16Z, looking at them, how do you get an investment thesis? These science is hard. You're doing this really early bets on something that's science and biology that's harder to bet on. So how do you come up with this investment thesis? How do you get your mind around so many different threads of technology, science, chemistry, biology? And yeah, I would love to know more about that.
Yeah, absolutely. So I think the fundamental thesis that A16Z Bio was started on, which we were started about 7, maybe 8 years ago now, by Vijay Pandey, who was a professor at Stanford, who really had, I think the early insight that now many others are jumping on board with as well, that there is this amazing intersection that's happening at the intersection of engineering, computation, and biology. And like you said, that we're getting to this place where we can digitize biology, essentially, because of the fact that DNA sequencing and DNA synthesis have gotten so inexpensive, so routine, so high throughput. We're able to read out what prior was more of an analog type of analysis of biology. We can now digitize and do that on a very high scale. And that combined with advances that are happening in the tech world, in computational power, in AI and ML, allows you to then interpret that data. And so it's this convergence that's happening at the ability, as you said, to read biology, to interpret it, and then to test that, to write it and test it in a true design, build, test loop that's really allowing us to iterate in a way that wasn't possible before and understand biology on a much larger scale than was possible before. So that, I would say, is what the fund was started around, this thesis. And so those tend to be the types of companies that we're interested in. But that's really expanded over time. So we invest now in everything. The bio and health world expands from everything from tools and technologies that allow us to do those things. So new proteomics technologies, new DNA sequencing or synthesis technologies, to platforms for discovering targets, for developing new drugs, for bringing those drugs forward to the clinic, for identifying the patients that they would be most specifically appropriate for, to digital health, and then healthcare delivery. So once we actually make these drugs, how do we actually bring them to patients? What are the technologies that could allow doctors to do their job more efficiently? We also do some investments in the synthetic biology outside of therapeutics world, so think the next versions of Impossible Foods. But to your question more specifically around how we develop those theses, first of all, we don't get any sleep. Second of all, the team. So we have a really fantastic team that we've built out that are folks who do have a lot of depth in specific scientific areas, but are also people who are really interested and able to move broadly and to expand upon the places where, for example, they did their PhD. So within our team, one of the things that we like to do is spend time going over all the areas, maybe for that year, that we think are really compelling, that we'd love to dive into, and then divide them up such that each of us tackles some, goes really deep into it. So talks with professors, talks with people who are developing companies in this space, understand what the KOLs think, understand what is being done, and then spend time to actually think about what would a company in that space need to look like? What are the 5 criteria that perhaps we think would make an investable company in that space? And then we spend time as a group sharing that information with each other so that we all can learn from those areas that we've gone deep. We also lean really heavily on our portfolio companies. So we invest in people, and we think those people are fantastic. And so I spend a lot of time with CEOs or CSOs or CTOs of our portfolio companies, understanding what they think is interesting, what they're seeing that's coming up next. We've also built out a really fantastic advisory network of folks, both from industry as well as from academia, to lean on them to understand what they're seeing as well, because we're not going to be the world's expert on everything, and I think it takes a village. But fundamentally, we're looking for companies where, first of all, the idea is big. Like, if this works, will anyone care? This can't just be a step change. It needs to be something that is a big enough idea that it will fundamentally change what is possible in a field. And then I would say the second question is like, are these the right people to do it? And those are kind of the two big questions that all the other criteria, all the other investment theses build upon.
I see. That's very useful, because while we are ideating on ideas in the space, we are a lot of us have the tech background coming in, and we've built like tech platforms, and some of us have dabbled in biology a little bit, and so we are trying to think, what are some areas that we should look into? CRISPR is big at the moment. mRNA, the vaccine was a big thing. LLMs, we have a huge community here of enthusiasts who are actually building LLMs for specific areas, and there's a lot of work going into creating LLMs specifically for biology now. While we're on that topic, what are some of like key technologies or key areas that interest you the most? And it could be companies you've invested in, and please feel free to share.
Sure. Yeah. So if you guys are working on LLMs for biology, I want to talk to you. Because I think that, just to that note, well, I can talk about this because it's an area I'm interested in. We've gotten to a place today where, at a really respectable way, we're able to, from sequence, be able to predict structure. But I think we're moving to a place where, from structure and from sequence we can predict structure, and from structure we can predict function. And if we can do that, then we can work backwards and say, what is the function that we want to achieve, and design that in a generative fashion. And that, I think, is going to be a huge step change that is coming. So some of you maybe are building in that space. So areas that I'm excited about. So you talked about mRNAs, you talked about gene editing. Those are really important spaces. Both of them are limited by something called delivery. So I think delivery is one of the huge nuts that needs to be cracked in the next decade. We have all of these incredible new modalities that can do amazing things to engineer cells, to introduce new functions, but we fundamentally are limited by our ability to deliver those to the right types of cells where they need to have that function in a safe way. And most of us have been injected by lipid nanoparticles, LNPs with our COVID vaccines, but the reality is it's a very crude version that really has no specific direction to one cell type within your body, and works because for a vaccine that's okay. But there are really incredible things that we could do with mRNA technology, for example, with gene editing, with CRISPR, if we could only deliver it to specific cell types in your body. So that's one area that I think is really interesting, and where I've actually seen some compelling work in recent days applying AI and ML to predict new delivery technologies that could deliver to specific cell types. So that's one area that I think is really interesting. Within that delivery bucket, I'll highlight one of our portfolio companies that I think is doing really exciting work in that space, and that's a company called Dino Therapeutics. So they're located in Cambridge, and this is work coming out of George Church's lab. The founder, Eric Kelsig, was in the Church lab. And the fundamental idea here is so there's this one kind of delivery methodology or technology called AAVs, or adeno-associated viruses. And this is a virus that exists in nature and that many of us have actually been infected by and had no idea. And people have been using AAV for a while to deliver gene therapies. AAVs, though, like most delivery vehicles, tend to go to the liver, and it's hard to get them to go to other tissue types. And people for a long time have been trying to engineer AAVs to go to new cell types. And they would do that by what they called capsid shuffling. So essentially, you would take the genome that comprises AAV, you would cut it up into pieces, you would reshuffle it, and then you would test in mice or in human cells and see, with this shuffling, where does it now go? But the reality is that wasn't a smart way to do it. It was just relying on random chance. And so what Eric Kelsig did, and this work turned into Dino-Therapeutics, was say, instead, let's go through the entire genome of AAV, and let's make single amino acid mutations, and then let's see what those single amino acid mutations, how they affect the ability to infect specific cells in the body, the ability of the AAV capsid itself to form, the safety, so does your immune system recognize this, the ability to redose, the ability to manufacture, all the characteristics that you care about. Let's, in a very high throughput way, map out how each of these amino acids affects those characteristics. Then let's start to do that in combination, and then let's apply ML to those data sets and now start to predict what combinations would achieve what characteristics you're looking to build in your delivery vector. And so that, I think, very much fits in with exactly what we were talking about before, the ability to do high throughput screening, the ability to do this in a, to use DNA sequencing to digitize this output and do it in a much higher scale, and then apply computation such that you're not now screening the same libraries, you're designing new libraries that didn't actually exist before in nature in a smart way instead of in a low throughput, dumb way. So that's one that I think is super compelling. Another portfolio company that I think is doing really cool work and is very much aligned in the areas we've been talking about is in Citro. So in Citro is in the Bay Area. This is work coming from Daphne Kohler, who was the chief computation officer at Calico, had started and was the CEO of Coursera and was at Stanford previously. And the idea here really is to use, again, high throughput assays on cells that have been taken from human samples and collect a variety of different data types. So whether that's RNA sequencing, whether that is images of the cell types, whether that is proteomic readouts, and then, and so these are disease cells that came from patients. And then make a series of perturbations to those cells via changing the genetics, maybe adding small molecule drugs, and see how that changes the way that the cells read out in all of those different assays, and then apply computation to that data and try to make sense of it. So essentially try to map disease biology and then figure out how we can alter disease biology via the drug modalities that we have existing here today. And then I'll highlight one more company because I think, to be fair, I should highlight one that's not a portfolio company. So one company that I think is doing super cool work is Icon Therapeutics, also in the Bay Area. This is a company based on work from Bob Tejan and a variety of other faculty members from UC Berkeley. And they essentially developed this really cool new super form of microscopy. It used to be that when you image cells, first of all, it wasn't super high resolution. And second of all, you were imaging them in a static fashion, such that you only got a snapshot of where the cell was at any given time. They've developed a way that you can track single molecules within cells, within live cells, over time. And so what's really cool about this is instead of adding a drug to a cell and then saying, okay, what happens to the overall cell? Does it die? Does it live? They add drugs to a cell, and then they go in with these amazing microscopes and computation and watch what happens to single molecules. Does it inhibit the interaction of this molecule and this molecule? Does it change where this protein is located within the cell? Or maybe even the dynamics of how it gets there? And so I think that is super cool.
Yeah.
Yeah.
Yeah. You just mentioned LLMs, and if someone's working on LLMs, we have a bunch of people in our group who are, and the LLMs channel, we had, just before this bio series, we had the LLM series. And about 40, 45-ish people participated, and it was a super hit. And some companies came out of just jamming on ideas, but that channel tonight is going to be very busy.
Oh, awesome. Can't wait to hear.
Yeah. Becky, if we may move to what you do at A16Z, which is technology platforms for therapeutic discovery. And personally, I felt it was a very interesting way to look at how drug discovery is done now, where you have pieces of that you go deeper in and you keep it as a platform, the delivery mechanism, the initial finding hits mechanism. Whereas 20 years ago when I was doing drug discovery, it was like, you start here, you form teams as you go along at different phases of the project, then you have toxicity studies, and then you dismantle the team, and then you start all over again for different drugs. But this is a very high throughput way of scaling each part. So do you mind giving us more insights into how the drugs were previously done, and how this thinking about it as a platform has changed the way of how drug discovery is done today?
Yeah. I mean, look, I think to be fair, if we go back to like the birthplace of biotechnology, which was here in the Bay Area, at least we'll claim it, many people will credit the birth of biotechnology as a industry to be when Herb Boyer at UCSF essentially discovered that you could take enzymes and you could cut up pieces of DNA with them, and you could recombine those pieces of DNA, put them back into bacteria, express a protein from it, and ask the question, how does changing DNA sequence change a protein? He then teamed up with Bob Swanson of EC and started Genentech. And so I think of Genentech as like the OG TechBio company, because that was a platform. They weren't based on some specific program that they were trying to advance. They were based on developing the platform of like, how can recombinant DNA change what's possible for our understanding of proteins and enzymes, and then develop that into ultimate drugs, right? And so I say that because I think this is an evolution. This is a course that we've been on for many years. This platform biotech is not a new phenomenon per se. I think that new advances in computation and this digitization of biology that I was talking about have enabled platforms to develop out in a way that perhaps wasn't possible before or at a larger scale, and have allowed companies to then capitalize on that and be able to take that data, take that learning, continue to iterate on it, and continue to develop programs from that platform. But I will also say that especially in today's funding environment, companies are being judged on their products at later stages. And so even for our companies that are, all of our companies really are platform companies that are based on a novel technology that allows you to continue to learn, continue to iterate, continue to develop new biological insights. At some point, that needs to be translated into a product, whether that's a drug or something else, that can stand up by itself, can give value to patients. And so I think that is one of the biggest challenges for companies in this kind of tech bio space right now, that is, where's the proof of this working, Because a platform is fantastic, but you need to show that that platform actually lays eggs eventually, right? I don't know if that answered your question.
Yes, it did. We had a session yesterday on the TechBio series.
Yes.
And one of the first questions that Mitra asked all of us was, how do you define TechBio? And there were a lot of answers. It should be TechBio, it should be biotech. No, it should be health tech. Oh, you should be opinionated in your biology. then it's real biology. one of my answers was when we were trying to raise money for the previous startup that I was a part of, we used to joke, and it's partly true, I guess, that if you say biotech, you don't get the multiples that when you say tech bio, because all of a sudden you have tech coming into bio, and tech is nimble. So I want you to take a side, and I want you to have a spicy take on this. What is your opinion? What is it? Is it tech bio? Is it biotech? Is it health tech? What is it?
So, first of all, I will say I don't like the term tech bio. I'm glad I have some supporters in the audience. There are several reasons for that. I think one of the fundamental reasons for me is I see them as concentric circles rather than a Venn diagram, right? I think like all tech bio companies are biotech companies, but not all biotech companies are tech bio companies. I don't know if that makes sense to you all.
I'm confused.
But, okay, all right. So we talked about Genentech being the OG biotech company, right? Over time, the Genentechs of the world, the Amgens of the world, many other of those companies have perhaps slowed down in some ways in innovation and have become more focused around the assets than around the platform or the technologies. And so I think there are many examples of biotech companies that are focused on one target, let's say. They have made an insight into this amazing protein in the body and have a very clear insight on how to drug it. That's a biotech company, They are taking advantage of modern technologies to develop a drug against that one target. I don't see that as being a tech bio company because fundamentally I see tech bio companies as those that are taking advantage of what we talked about before, this digitization of biology, this ability to, in a high throughput fashion, understand an area of biology more thoroughly, and continue to learn from that, continue to iterate on it, such that the knowledge we have today and the platform we have today is not the knowledge and the platform that company will have in 10 years. And you'll continue to be able to make insights, continue to be able to make novel drugs or whatever it is that your product is because you're learning from data rather than staying stagnant in time, if that makes sense. And so there are many biotech companies that are fundamentally not a tech bio company. But every tech bio company, I think, is a biotech company. And my worry by trying to separate the two is that you actually cheapen what's being done in tech bio. I think that tech bio is a very serious area with serious people who want to make products, who don't just want to do an AI for X, Y, and Z and have a cool headline and not actually bring something to patients. Mm-hmm. And so let's be serious about it. Like, we are not different. We're just better? No. No. we're just a subset that is focused on a fundamentally different approach. So that's why I don't ever use the word tech bio really, because of the fact that I feel like it cheapens the work that's being done. So health tech is different though. And health tech I think is a completely separate sector than biotech in my opinion.
So is that a +1 for biotech from Becky?
Plus 1,000 for biotech from Becky.
Okay.
Yeah.
Okay.
Yeah.
Sounds good.
But I totally get why people like to kind of come up with new language, new terms, take ownership over what we're doing. It feels like it builds a community. So I have many friends who use the word tech bio, and we will still remain friends.
So within the community, what we've observed is giving it the term tech bio makes it less scary for people who are outside and not from the bio community. Mm-hmm. And it feels like I'm a techie and I can possibly help in the bio space. So maybe a couple more questions before we open up to the audience. One of the questions we have in the community that comes up over and over again is, I'm an engineer, I've built platforms. I'm an engineer, I've scaled backend systems. The COVID pandemic has changed my perspective. about what's important. I want to contribute towards biology, but I don't know where I can start. Do I need a lot of domain knowledge? Do I need to know, do I need to brush up and go back to taking biology courses or chemistry courses for that matter? What would you say to such people? Where can they start, or what are some areas that they could start making an impact and then go deeper into the biotech space?
Yeah, it's a good question. I would say so I'm a biologist, and I am not as deep. I do what I can, but I am not as deep on the computation side. And I think one of the amazing things about TechBio companies, biotech companies, is that fundamentally they tend to be very diverse in the types of different areas of expertise, the types of different functional groups that are brought together. So one of the hallmarks, I think, of these types of companies is they tend to have folks who are chemists and biologists and computer scientists and structural folks and machine learning and people who bring in manufacturing and a variety of different areas of expertise. And none of those people are ever going to be experts in all the other areas. Fundamentally, what you do have to be able to do is communicate with each other. And so I think that's probably the biggest barrier for folks, is being able to start speaking the same language. And so look, I think, first of all, there's a lot of great resources available today. For example, there's a blog series that started last year called, this is funny actually, it was called Decoding TechBio. And just last week they changed it to Decoding Bio because of exactly the conversation we just had. But this is the purpose of this group. It's written by a great group of friends, is to do exactly what you were just saying and bring together the folks on the tech side, the folks on the bio side, help communicate between the two groups what's being done and form common language. And so I would say, don't be scared. You don't need a PhD in biology. I have a friend actually who's a pure computation person who recently told me that they hired an undergrad as a tutor because he was like, I wanted to learn just enough biology to have a conversation. And so I figured, look, this undergrad is currently taking these classes, he can just share to me what he's learning. But I think that there's so much happening right now in terms of communication on YouTube and these types of conversations, that just starting to meet people and have conversations about what's happening in other areas is a good way to start. And I think it also really depends on what type of company you end up working with. So for example, if you're in a computational chemistry company versus a company that's maybe doing computation for image analysis, the types of science that you need to maybe understand and be conversant on are different. And so I'd say start having conversations with biologists and ask them what they're interested in, where they think computation could take or engineering could take a really important role, and learn from that rather than trying to dive too deep without knowing where you're going. I don't know if that's helpful, but yeah.
Yep. We have time for maybe one more question before we open up to the audience. So this is, if a genie came to you and granted you a wish to change anything about this space, a hard problem, or I just wish, from what we were just talking about, I just wish I could figure out in advance how to deliver this drug to this tissue or to this cell type. What is that one key technology that, in a way, trying to ask, what is that one key technology that you're keeping track of, and he was like, that's the one I'm putting my bet on, I really feel strongly about, and I hope in the next 5 years it breaks the laws.
Yeah. So I already talked about delivery, which I think definitely fits that category. But I'm actually going to go outside of a technology, since you gave me my genie lamp. The thing that worries me most is that these early stage technologies that we're working on that have huge impact to actually cure disease and not just ameliorate it, that they'll never actually be able to reach patients, because by the time we get this crazy gene therapy approved, it's going to cost, $3.5 million, and 50 rich people in the United States are going to be able to afford it, and no one outside of the United States will ever see it, applied to them. And that is one of the things, honestly, that worries me most, is like, back to the original conversation that we had of like, why did I move outside of academia? To make an impact and bring curative, impactful, meaningful medicines to people. And I don't just mean rich elite people in the United States. And so that is one of the things that I think we really need to get ahead of and figure out, like, how do we work with payers and providers and regulatory agencies to make sure that there is space for the innovative therapies that we're developing today, and that there's a structure for them to actually be paid for and be manufactured in such a way that they can reach everyone who needs them.
Thank you. So with that, I'll open it to the audience. Mitra is carrying the mic here.
Oh, thank you. I had a question I was really excited to ask, and you answered about 70% of it. So I'm gonna try to ask the interesting part of the remainder.
Okay.
It's about a gadfly position that comes up about TechBio, which is that a lot of the value proposition of TechBio is about accelerating the drug development process. AI and ML are going to make it easier to identify targets and build molecules. High content screening and high throughput assays are going to accelerate preclinical work. Simulation is going to tell us how best to run our trials, all right? So the idea is things are going to go faster. At this point, the idea of TechBio is 8 or 9 years old. At what point is the thesis falsified? At what point has it not happened faster? Is it too soon to say? Is it a fair criticism? I just kind of want to open the floor and hear what you have to say about it.
Yeah, that's a good question. I personally don't think that is ever going to happen, that it's going to be falsified. First of all, it takes time to be able to actually measure what you're saying, because for drugs, right, you need to be able to actually take these things into the clinic and then prove that they work and then get them commercialized and all of this, right? And then you also have to do that in such a time scale that you can retroactively go back and answer the question of how fast might this have happened without TechBio, without application of computation. And I don't know if we'll really ever be able to do that, because, like I said, this isn't something that started 7 years ago. This is something that has been building over the last decade plus. And so, if you talk to chemists, for example, they'll tell you, yeah, we've been trying to apply computation and predictive modeling to our medicinal chemistry experts for a while, and it's been getting better and better over time. But it's not like there's been this switch from zero utility to now jumping off the deep end and being full on board. So I think that's going to be a hard assessment to make. What I'm interested in is when we're not at a place where we're just speeding up the process. Speeding up the process is important. But I think the most important thing is increasing the likelihood of success. Because if you make it there faster, that's great. But what we don't want to do is spend a bunch of money putting drugs into people that we could have predicted weren't going to work based on data. And then the other thing I'm excited about is places where we can predict new molecules, new strategies that we simply wouldn't have come across by happenstance. So if we go back to the dino therapeutics example, why I think that was so exciting is because by simply reshuffling existing AAV sequences, we weren't going to get to a place that Dino has reached where they can say, let's design new sequences that simply wouldn't exist in that reshuffling. And so I think it's more of that generative aspect that's really going to be compelling for this industry. Yeah.
Hey, how you doing, Becky? Very good talk. Question for you is, what would you say is the current state of simulations for cells?
Simulations for cells. Ooh, that is a very good question. Simulations for cells is a much, much, much more difficult thing than simulations for single proteins, obviously, because of all of the different components that go into a cell. I think that's one of the reasons why when I mentioned ICON, why I think their technology is so interesting, because on a larger cellular context, you're able to see what's happening and take that data to be able to make some predictive modeling. I personally haven't seen any good examples of simulations that I would trust on the entire cellular level. I don't know if anybody else in this room has, but I think that's actually a fascinating idea. I like it.
Cool.
Yeah, yeah.
Thanks for coming out to give this talk. I'm curious about something you mentioned at the beginning, where when you were at Big Pharma, you saw how hard it was to partner with startups, and now you've kind of seen the other side of it, working with startups, trying to partner with Big Pharma. And I'm curious, why is it so hard, and what advice do you have for startups who are looking to partner?
That is a great idea, or a great question, and one of the things that, when I make investments, I oftentimes put on kind of the lens of a pharma BD person, because for many of these companies, their initial go-to-market will be via a partnership rather than them taking their own program forward. They may eventually, but so I always ask myself, okay, who would partner with this company and how much would they pay? To your question of why it's so hard, it's different for every organization, but one of the reasons oftentimes has to do with actually just structuring of budgets. So many big pharma companies may have massive piles of money set aside to be able to do these large asset in licensing deals, but if it's a smaller collaboration, oftentimes you'll actually be asking the R&D team to take away from their own budget that they've set aside for their own R&D programs and shift it over to this collaboration. And so that's like asking someone to give up their baby for some adopted child they've never met before, and may not work, and and so it's just not a fair trade-off to ask for. So that's part of it. Even if that budget constraint doesn't exist, there's always a time constraint. And so for these partnerships, usually they're true partnerships. It's not like you're outsourcing everything to the small biotech company. Usually, like I said, it's a situation where the pharma company is bringing something to the table, the biotech company is bringing something to the table, and hopefully you've got a joint research team who's iterating back and forth. And so they have to then give their time to be able to do that also. And so it's, I think, just a resource constraint, whether that resource is time or that resource is capital. And so pharma companies always have to be making risk assessments, and oftentimes their risk assessment for early stage technologies is fundamentally it's less risky for us to just wait and see if this works, and if it works in a couple years, then we'll pay up, or then we'll buy them, or whatever. And that might be true, but it's a whole lot less interesting and fun. So, and then your advice on kind of how to help. I think one of the big things, so one thing that we have, I think that's a really great resource at A16Z, is we have a business development team, and their job is to meet with the BD folks, the R&D heads, multiple different touch points along the organizations at all of the pharma companies and mid-stage biotech companies, and to keep up to breast on what are they interested in, what types of deals do they do, exactly what person is responsible for championing deals, so that when we have a company who's interested in doing partnerships with biopharma, we can say, aha, you should talk to Pfizer, Novartis, and Biogen, and these are the people you should talk to. Oh, and I actually know them well and I can make an introduction, because ultimately people do deals with people. And so it's important to like form a relationship with not only the BD people, because that's going to be important to get a deal done, but also form a relationship with the scientists, because the best way to get a deal done is if you as a scientist at a startup company convince a scientist at that biopharma company that this is worth their time and it's worth their budget, and they're willing to go advocate to their leadership and say, yes, I think this is a good idea and I'm going to vouch like my reputation on it. So relationships at the end of the day. Yeah.
I think we said me next to you.
Hi there, thanks. So I feel like a lot of these companies are generating so much data, right? Imaging data and genomic data, like 23andMe is sitting on 15 million genotype customer data, right? So let's use the example of Dino, where they're generating data and they could predict that using AAV, you can deliver it to the heart instead of the liver. But then you go into clinical study, and what if that's not true? Then do you say, is this a data problem, or is this an AI ML problem? Is it the quality of the data that's not good, or is it the quantity of the data that's not good? Like, when do you have that reckoning where you say, generating more data isn't actually predictive?
So I think this is going to be a huge thing going forward. It's not about more data. It is about the quality of the data, where the data comes from, how it's collected, all of these things. So in your example of translatability of delivery technologies, we are already starting to see whether these things were designed by old school methods or by AI and ML, that delivery technologies don't translate oftentimes from mouse to monkey and from monkey to human. And so that's not a problem that like the data was collected incorrectly. It's a biology problem that fundamentally the biology of these different species is not exactly the same, and therefore the data that you've collected in one is not 100% translatable. Now, maybe some of that could be fixed or ameliorated by computational methods. Like if we could understand how the biology is different from a mouse to a monkey to a human, maybe we could better interpolate the data that we'd collected from a mouse or from a monkey, for example, and predict how that might be different once you move it into a human. But fundamentally, I think one of the big bottlenecks that has happened in application of computational methods up until recently has been the data that we're using was simply not collected with this purpose in mind. So for example, I remember when I was at Amgen and we were doing, like partnerships with companies that had built out new, AI, ML based methods to be able to analyze essentially chemistry. And one of the problems we discovered as we went back through all of our old historical small molecule data was it was collected in such a way that like it was different people pipetting, different assays that were set up slightly differently. And so, dirty data simply isn't going to ever allow you to build predictive models that are useful. And so I think we need to be, and that's actually, I think, a great way, back to your question on this intersection between engineers and biologists. One of the things that engineers and computer scientists can help biologists with most is helping them understand how to better set up their experiments. Like every biologist before they set up an experiment should have to sit down with the bioinformatician who's going to help them with it and say, okay, this is what I'm trying to do. This is my goal. How do I need to set up this experiment such that at the end of the day, you feel comfortable with the data that's collected? What types of controls need to be put in? What types of replicates do we need to be able to power this experiment? And that's true at the early experimental level, but like that also needs to then lay out, I'd say translate into the clinic as well. So the way that we design clinical studies needs to be fundamentally changed such that the data we're collecting is compatible with these types of computational analyses.
I think we have time
Just-
for one more question. Why don't you pass it, and then we can break and people can continue chatting on that.
Just a thought around there. In my experience, when I have worked with, as a computational biologist, I've worked with scientists, planning that experiment and planning why we are doing something together really helps you also in your communication, because ultimately the data comes back to you, and if you don't know why you are doing something, that's a huge loop that is very useful. And then you also develop this rapport with the lab scientist, and if something doesn't quite work as the way you expected with your hypotheses, you can always say, be sure the experiment was planned correctly. Okay, what could have gone wrong? So it's a very vital thing. I completely agree.
You spoke super briefly about how we want to have medicines and treatments that are accessible beyond the US and beyond kind of the upper echelons of society. Do you think that's possible in the current healthcare system with regulations and with insurers? If so, what's blocking us and what needs to be done? And if not, what are you, or A16Z, what are you doing to actively invest in changing the system, evolving the system such that we can reach a point where there's equity kind of across therapies?
Yeah. So is it possible today? It depends on what type of drug you're making. So I think if you're working with a modality such as like a small molecule or a biologic, then yes. There are really unfortunate regulations that have been implemented at the highest of governmental levels, such as the IRA, which some of you might be aware of, which essentially is like disincentivizing some of the innovation that's happening in small molecule therapeutics. But I think doubling down on modalities like small molecules that are easily manufacturable at bulk, where the costs are low enough that you can, in a realistic way, be able to distribute them throughout the world. We think that it's really still important that there's going to be a resurgence of old modalities, of small molecules, that we can now, apply new methodologies, whether that's computation or novel ways of screening, to innovate on those old modalities. So I think it's important to double down on that. I don't think today that, given the current way that both manufacturing happens and the way that essentially the entire incentive structure is laid out for biotech companies, that these types of really expensive novel medicines are going to be accessible to the broader world today. And that's because they're simply so novel and so expensive to make. And that part of that is clinical development. Part of that, in the example of, for example, cell therapies, is simply like the actual manufacturing costs of generating the drug product. So today, no, I don't think that it's possible. How do we get there? There's a couple different things. So we actually just recently hired on someone at the A16Z team whose job is going to be our advocate between the biotech industry and between A16Z and the government and the FDA, and whose job is going to be to go out there and make change and set the policies that our companies are going to need to be able to bring their products to the market in a sustainable way across the world, even if that's not for 10 years. In 10 years from now, we think that we need to be proactive about that as investors and as innovators in the space. So that I think is really important. And then also investing in things like new manufacturing technologies that allow you to produce these medicines in a less costly, less cumbersome manner. But it's a real problem, and I think there's no simple solution for it.
Please give a big round of applause for Becky.
Thank you.
Thank you.